everybody, and welcome to Chapter 9. This is going to be Sexual Reproduction and Meiosis. This chapter is going to discuss the production of sperm and egg cells. In a previous chapter, we talked about mitosis, which was the production of what's called somatic cells. So in this chapter, we're going to be talking about the production of sex cells. Somatic cells are your typical cells like skin cell, liver cell, a stomach cell. These cells, sperm and egg cells, these are sex cells. They are different than our typical cells. They have less chromosomes. They're genetically unique. When we replaced our somatic cells, like our skin and liver cells, they were an exact replica of the parent cell. So we're going to hear some similar terminology in the processes, but there are going to be some major differences. So meiosis and mitosis, it sounds familiar, but it is two different processes, and it's very important that you understand the difference between these two processes. All right, so the first thing we're going to talk about is asexual reproduction briefly. An A, when you put an A in front of something, that means no or not. With asexual reproduction, it's rare. It's usually unicellular organisms. Think of like bacteria. And basically, the offspring is a exact replica of the parent. Whereas a multicellular organisms, they have two parents that produce, one produces, the male will produce a sperm, the female will produce an egg, they come together, and so you get a genetically unique offspring. So there are some exceptions. There are a couple multicellular organisms that can do asexual reproduction, like sea stars, and there's some worms, but it's very rare. Just briefly discussing asexual reproduction here. You can see this is some sort of unicellular organism, and basically the offspring just basically buds off of that parent. And for sexual reproduction, we're going to be using the term meiosis. And this is a type of cell division that's going to be forming either a sperm in a male or an egg in a female. If you go wandering around the internet, you may see some terms called a spermatocyte or spermatid, or an ovum or an oocyte. Those are some little bit higher level terms. We're going to be sticking with sperm and egg. So each of these is genetically unique, and then when they come together, they're going to be producing an offspring which is very genetically diverse, okay? And so that's important, okay? That is important because genetic diversity, later on we'll talk about how that plays a role in evolution and natural selection. And so it requires a lot of energy to have the sperm and the egg and develop the offspring. It requires either like an egg or a placenta for the offspring develops, but it does allow genetic diversity. All right, let you guys read through that. Okay, so as you know, our cells contain chromosomes, okay? And there is a phase, as we've discussed in the previous chapter, called interphase. And during interphase, before the cell division, you're going to be replicating your chromosomes. So you can imagine that you have one from mom. Oh, let me get my pen going here. You have one from mom and you have one from dad. And what's going to happen is they are going to replicate. So there is the copy of mom's and there is the copy of dad's. And so they are held together by a what's called centromere. These are all terminology that was in the previous chapter. And so now you have these duplicated chromosomes, okay? And the duplications are called sister chromatids, but they are a homologous pair. So this could be chromosome number 2 or chromosome number 4. In a normal cell, like a skin cell or a liver cell, we are going to have 23 pairs of chromosomes. So one from mom and one from dad, or you can say a grand total of 46. Now here, what we're looking at, these are duplicated chromosomes. So this is what would happen in the S phase of interphase. When you do DNA replication, when you make a copy of the chromosome, you get these sister chromatids. All right, so we use the term diploid. Di actually is a prefix for meaning two. So diploid cells contain two sets of homologous chromosomes here. Okay, so you get one from mom and you get one from dad. So you can see here we have our 23 pairs of chromosomes. We have our 23 pairs of chromosomes. And so these are normal, these are not sex cells. So a sex cell is actually, we use the term haploid. Here we have diploid cells. So this could be a skin cell, like I said, or a liver cell, etc. And so looking at this right here, this is actually what's called a karyotype, which the word is used in the just go right to this next slide here. And so a karyotype is, it basically takes like microscopy, light microscope, and it takes camera and special fluorescence used. And you're basically taking a picture of all of your chromosomes, and they're able to order it in its order. So this is chromosome 1 and this is chromosome 14. So when you look at, like, there's be disorders that we'll talk about at the end of this chapter, like trisomy 21, there would be an extra chromosome because tri means 3. Now normally, like I said, you have one from mom and one from dad for every chromosome. So you have a pair. Okay, you have a set. These are actually called homologous chromosomes. Even these ones are also called homologous chromosomes, even though you have a copy of it. So it can either be this, or it could be this. This one's from mom and this one's from dad. So the term homologous chromosomes can also be referencing just the one from mom and one from dad, or here we're looking at one from mom, one from dad, and also its copy too as well. So we look at a karyotype. The first 22 pairs are called autosomes. And then the last pair is called sex chromosomes. And so if you are a female, you are XX. If you are a male, you are XY. You can see the Y chromosome is a lot smaller. There's not much information on the Y chromosome. For example, both males and females have an X chromosome, so there'll be things like color vision, blood clotting genes on there. Whereas the Y chromosome, since a female doesn't have it, and it just has male directed type of genes, so like testosterone, descending of the testes, things that a female wouldn't need. Besides, that kind of thing, males and females are very similar. They're almost exactly the same. So the 23rd pair is going to be your sex chromosomes. The 22 other pairs are called autosomes. And so you can see here again, this picture here, we have a set of replicated chromosomes right here. Homologous chromosomes, so if this is chromosome number 2, this one's from mom, this one's from dad. This gene is a gene for hair color, and this gene is a gene for hair color. This is a gene for eye color, and this is a gene for eye color. Now, if you're looking right here, you're probably like, oh, this one has a capital B and this one has a lowercase b, because there can be different variations of that gene, and that's something that we will discuss in the next chapter. So for example, this right here could be brown eyes and this one right here could be blue eyes, but it's still a gene for eye color, even though there can be variations of it. All right. And so going into this next term right here, so I didn't realize this was the next slide here, alleles are those different variations. So if this is, let's say capital A represents attached or attached earlobes, both of these parents have attached earlobes instead of unattached earlobes. Whereas if this is eye color, like I had described before, this parent may have brown eyes, this parent may have blue eyes. This gene over here could be for like hair color. You can see that they both have the lowercase, so it's the same hair color. So alleles are those different variations. They're the definition of an allele is an alternative version of a gene. So it's like blue eyes versus brown eyes, straight hair versus curly hair, brown hair versus black hair, attached earlobes, unattached earlobes. Okay, so we talk about in general how mitosis and meiosis sort of intertwine. They are part of what's called the life cycle. So you can see here we have gamete formation. So gametes is a fancy word. It's kind of just like a category term for a sperm and an egg. And so the process of meiosis will allow us to produce the sperm or the egg. And what you can see here is that you do not have a set of homologous chromosomes. Your gametes, they are haploid, which means they only contain a set of 23 chromosomes. They do not have 23 pairs. And that's important because you can see here the sperm which has 23 chromosomes, and the egg here, which has 23 chromosomes, will come together, and that will give you your 23 pairs or your 46 chromosomes. So meiosis is going to be the cell division process where we're going to reduce the chromosome number to single digits instead of double digits, right? I shouldn't say double digits, but we're going to go from pairs to not being paired, and we use the term haploid. Now, once the sperm and the egg come together, then you go through development. And so this is basically mitosis from here out. So you can see the fertilized cell is right here. And then from here on, we go through cell divisions to get to the human being. I mean, it's crazy. We start off as one cell, and that one cell just goes through a lot of cell divisions, a lot of mitosis cell divisions, and then we get our human being. So we all started from one cell. So both of these systems are required for each other, but meiosis is really only in the beginning, and then mitosis is throughout the rest of life. Okay, so we have what's called germ cells. Germ derives from the term germinate or to grow. And so basically we have these stem cells, these germ cells. And in a male, they're going to develop into sperm, and in a female, they're going to develop into eggs. And so these germ cells in the male live within the testes. The testes are basically little sperm factories, and the ovary in the female is going to be the little egg factories. So you can see at some point the sperm and egg cell will unite. Okay, so you can see sperm and egg cells are formed by meiosis, which is what we will talk about today. They will fertilize together. The fertilized egg is called a zygote. Be familiar with that term. And then once that zygote is formed, it'll go through a ton of cell divisions, giving us via mitosis to give us the human being, and then we become an adult. Gametes are haploid, which means that there is only 23 chromosomes. There are no pairs of chromosomes. So it reduces the chromosome number by half. And that's really important because if you can imagine, let's say these sperm and egg were like a typical cell and they had 23 pairs or 46. If they came together here, this would be 92. And then the next generation would be, 100 and something. And the next generation, and the next generation, and the next generation. So the chromosomes would just increase exponentially. And you can imagine all the errors that would be in there. So fertilization is the term for when the sperm and the egg come together, and when they come together they form that new organism. That is the first cell of life, and that is called the zygote. Zygotes are diploid. Diploid means that there is 23 pairs or 46 chromosomes. So that's why we use the term diploid, di meaning two. From there, that zygote will use mitosis to develop into a human being or an adult. Okay, so sperm carries 23 chromosomes, eggs carries 23 chromosomes. They fuse together, giving 46, so just like we had discussed. Let you guys read through those mastering questions. All right, so let's get into meiosis. So meiosis is similar to mitosis. There's going to be a lot of similar terminology, but one thing is there's going to be basically two rounds, okay? So if you remember mitosis had the prophase, metaphase, anaphase, telophase, and meiosis, we're going to have two rounds of that. So you're going to have prophase 1, metaphase 1, anaphase 1, telophase 1, and then you're going to go through prophase 2, metaphase 2, anaphase 2, telophase 2. And so that is something that's going to happen during that process. Some things that are similar is interphase will be there. Interphase only occurs at the beginning of meiosis 1, and cytokinesis will be at the end of telophase for both rounds. So there's only one interphase. So how is it different from mitosis? 2 divisions. It will end up with 4 cells, each having half the chromosome of the parent cell. So you can see here the parent cell, this is the germ cell. This is the germ cell, or it's really just a stem cell, and that germ cell is a diploid. It's a diploid cell, so it has 46 chromosomes or 23 pairs. And basically will undergo meiosis 1, meiosis 2, and you will end up with 4 haploid cells. 4 haploid cells. Each of those haploid cells will have only 23 chromosomes or a single set of chromosomes. We will also talk about genetic diversity that meiosis. If you look at some of these X's here, these chromosomes, you can see some of them have blue feet or pink feet, and that is the exchange of genetic material that we'll talk about, something called crossing over later on. All right, it's very important that you have understood mitosis before we move on to meiosis. So if you're still a little bit hesitant on mitosis or not really understanding it, at this point I would stop this video, watch either my previous video or some supplemental videos, because mitosis is very easy to understand. Meiosis, it uses the mitosis as a foundation, but if you just kind of start in here, you'll kind of get it a little bit confused in your head. So if you need to take a stopping point and go review mitosis, that might be a good idea at this point. All right, so meiosis, the DNA is going to replicate once. So it's going to do that in interphase. So before meiosis 1, remember meiosis has 2 rounds, meiosis 1 and meiosis 2, you're going to go through an interphase. So that's the same thing as mitosis. You have a G1, an S, and a G2 phase. In the S phase is where the DNA is going to replicate. And what's going to happen is your DNA replicates, and then you will not have an interphase before the second meiosis 2 round. But the nucleus is going to divide twice. And that's how we're going to get to a single set of chromosomes at the end of meiosis 2. But just looking right here at meiosis 1, I'm going to go ahead and zoom in here. If we look at this image right here, this is a nice overview of the events that happen. So here we have our interphase, and you can see that the DNA is going to replicate there. We have prophase 1. During prophase 1, you're going to see an event called crossing over, and that is going to be where we have an exchange of genetic material of the chromosomes. I like to think of like the chromosomes are wearing like hats, gloves, and shoes, and they basically just exchange pieces of their chromosome. So literally pieces of the chromosome break off, which can lead room for errors. I mean, can you imagine if like I give you my shoe and then you're about to give me your shoe, but it falls off and you lose it? Now you have a chromosome with a missing part of it. So some errors can happen. So crossing over is going to be a genetic diversity event that's going to occur in prophase 1. And so in prophase 1, just kind of remembering prophase from mitosis, you're going to see the mitotic spindle appear. You're going to see the nuclear membrane or nuclear envelope. It will eventually disappear at the end of prophase. In metaphase, you're going to see the chromosomes line up in the center of the equator. But here it is different than in mitosis, because here you can see the pairs line up. In normal, if this was normal, they would all line up and then break apart. So what happens is the copied pairs pair up. And how they pair up, this is going to be another genetic diverse event. It's called random assortment. So like there's here's mom on this side and there's a dad on this side. Well, guess what? Mom could be on this side and dad, it's just random. Okay, now anaphase, you're going to see the homologous pairs separate, whereas in mitosis, remember they would break, and this is hard for me to draw. In mitosis, they would break at the centromere, and so you would see that break apart and move. Here the pairs are moving apart, okay? And so this is something that's very, very unique to meiosis. So let's just go back to metaphase. In metaphase, instead of the chromosomes all lining up, the pairs line up. So this is the pair of the copied ones of the eye color. This is like chromosome pair 21, okay? Chromosome pair copy 21. So whatever side they line up on, it's random, okay? And so the pairs separate. Okay, you can see this is combining anaphase and telophase. So you can see you've got the reformation of the nuclear envelope. You've got cytokinesis. And now with meiosis 2, you're going to go through it all again. Now, meiosis 2 will be very similar to mitosis. It's very similar to mitosis. It'll be almost the exact, except the results are not, they're not the 23 pairs of chromosomes. They're just a single set of 23 chromosomes. But you can see here in metaphase, you can see now how the chromosomes line up, just like how they did in mitosis. So let's go through this, okay? Okay, so let's look at prophase 1. In prophase 1, the homologous chromosomes pair up and they form what's called a tetrad. So when they kind of form up and they exchange their shoes, which is called crossing over, they call that a tetrad. It's kind of like they link arms together. You can see the spindle fibers forming. You can see the nuclear envelope disappearing. Crossing over, you can see, is that where you exchange genetic pieces. Now this could leave room for error, meaning that part of the chromosome could break off and could not be replaced potentially. Now in metaphase 1, they're going to line up in the equator, but they line up as pairs. Okay, they line up as homologous pairs. And how they line up, it's called random assortment. So you can see this gives rise to genetically unique, and so this is what basically allows us to be different from, look different from our siblings, our brothers and our sisters. And so you can see how the chromosomes have lined up here, and you can see how they have lined up here. So you can see that the red one is now on this side, and you can see the blue one is now on that side. And that gives us different types of offspring here. So like I said, random assortment. I've also, if you start wandering around the internet, you may see what's called independent assortment. So some books use different terms, but it is the same thing right there. And so basically after anaphase 1, you can see that they have separated into different sides right here. And so you can look right here. Let's see. You can see how the red is on the right here, and over here it's on the left here. And you can see at the end, what kind of results that we have, and they are very much different. All right, so the spindle in anaphase 1 separates the homologous chromosomes, not the individual sister chromatids. So if you remember, your sister chromatids are, they have a centromere in the center, and in regular mitosis, this would have broken, and the individual sister chromatids. Here, the pairs separate. In telophase 1, those homologous pairs move to opposite sides. You're going to get reformation of the nuclear envelope or nuclear membrane. And the cytokinesis is going to split it into 2 separate cells. So after meiosis 1, okay, before we get into meiosis 2, we have 2 cells. All right, and so this cell is now going to go through meiosis 2. And this cell here is going to go through meiosis 2. Now, at the end of meiosis 1, this always confused me when I was learning this, is that we have 2 haploid cells, but remember, the chromosomes are still duplicated. So remember we had one from mom. Here, let me get my color changed here. And you had one from dad that was duplicated. So remember, these are, normally you would have something like this, right? Okay, but during interphase, we doubled up on our chromosomes. Remember, we made copies of it, right? And so normally in mitosis, the chromosomes would line up and then they would go their opposite ways, right? But what's happening here is that the pairs are lining up. Let me do it over here. Okay, and remember they did some crossing over, so they're going to have some different shoes on them, parts of them. Okay, and so what's happening here, this is in anaphase here, is that the pairs are separating. Okay, and so they use the term haploid. Okay, and you're reducing the number of chromosomes, but they use the term haploid because you have just one set of mom and one set of dad on each side, or one set from each parent on each side, right? Because this is going to be another cell, and this is going to be another cell at the end of telophase 1, right? But you still need to reduce that chromosome number to 23. So we're going to go through meiosis 2 after this, and that's in meiosis 2 where we're going to actually break apart the chromosomes, okay? And so this gets confusing because they use the term haploid at the end of meiosis 1, All right, but if you count the number, it almost seems like diploid, but we do have only one set of mom and one set of dad at the end of meiosis one on each side. So there's a set from, they're mixed up. Their genes are shuffled. So you have one set of chromosomes over here and you have one set of chromosomes over here. That's why they use the term haploid, but the chromosomes are still doubled up in sister chromatid form. So remember, these are sister chromatids here. All right, so in meiosis 2, it's exactly like mitosis, but there's no DNA replication before this, meaning that there's no interphase before this. So we take our cells that are our remnants from meiosis 1, okay? And so you can see that this cell is going here and this cell is going here. So we go through prophase 1. You can see that the nuclear envelope disappears. You've got the spindle formation. Here you can see the chromosomes are lined up on top of each other, not next to each other as we were in metaphase 1. You can see the chromosomes break apart in anaphase 2 at the centromere. They move to opposite ends, and at the end we are going to get 4 unique, so non-identical haploid daughter cells, all right? And so this one is going to have 23 chromosomes, this one's going to have 23 chromosomes, this one's going to have 23 chromosomes, and this one's going to have 23 chromosomes. If you are a male, these would be 4 sperm, and this sperm might carry an X, this sperm might carry a Y. So a sperm is technically what determines if you're going to be male or female, because a female will always give their offspring X's, because it's the only thing they can give their children, but a sperm can either give an X or a Y. So these individual sperm cells, some may carry an X chromosome. Remember, you only carry one set of chromosomes. So that 23rd chromosome is not a pair. It's either the X or the Y. I find it easier because a lot of times, like I have provided supplemental videos, I find it easier to watch some of the illustrated videos just to get a better idea rather than reading the words. That always helped me. So during meiosis, we're going to be reducing our number of chromosomes by half, okay? And so reading through that clicker question there, let me go through this one here. So what's nice about meiosis is that it creates this genetic variability. So looking at these puppies right here, you can see that they came from the same parents, but they have lots of variety of colors. And that was due to the shuffling of genes that happened in crossing over and random assortment. So crossing over was a big thing, and this is where you had the exchange. So this was in prophase 1. So crossing over, it's important that you understand that it occurs in prophase 1. You can see that these shuffling of genes have occurred here. So I like to say like they exchange their shoes. They're like, oh, I like your shoes. I'll take your shoes and I'll give you mine. And so now we have some chromosomes with some genetically unique. So it shuffles the genes, creates new combinations. We have what's called independent assortment or random assortment, and that's where the chromosomes align up. When they line up in metaphase 1, they line up as pairs. And how they line up can vary. So you can see in this alternative, you can see this pair is flipped right there. You can see here that they're just, all these different alterations are different. So it gives you cells that have very, very different results. One other thing that happens that creates genetic diversity is the random fertilization. So this is something that's unique, that's can be discussed a little bit in, not for this class, but you know how the result of meiosis 2 is 4 sperm. Well, females only have 1 egg, and they'll have these like 3 non-producing little things called polar bodies. That egg needs a lot of nourishment, so everything kind of goes towards that 1 egg. But males, hundreds of sperm, millions of sperm, and whichever sperm sort of unites with that egg is random. And even a female can't ovulate more than 1 egg, so which can be fertilized by another sperm giving you twins. So we have what's called monozygotic twins. We use the term mono. Mono means one, and that means there was 1 zygote. So the zygote, remember, is when the sperm and the egg fuse together. And basically what happened is it basically divided. like that zygote divided right away and it split into 2 embryos, which became 2 individuals. So these are identical twins. And identical twins is not genetic, to my knowledge. Like there may be special exceptions, but it's a fluke. It's basically a fluke, all right? Dizygotic twins, this is where you have the mother, usually a mother ovulates only 1 egg, whereas she may ovulated more than 1 egg, and you have a different type of sperm that fertilize. The egg is different from the other egg, and the sperm is different from the other sperm. So it's basically 2 fertilizations, I want to say different DNA, but genetically unique DNA. Okay, it's just like having dizygotic twins is like having an older brother and a younger sister. Like it could be that even though they were born at the same time, they are completely different and unique. I have a friend that has twins, and the daughter is blonde hair and blue eyes, and the son is dark black hair and dark brown eyes. So dizygotic twins, di meaning 2, you have 2 separate fertilization events here. This is genetic because this is dependent on the mother hyperovulating. The male always has tons of sperm, but the female, she's supposed to only ovulate 1 egg. So if she is a hyperovulator, meaning that she ovulated 2 eggs, which is carried down on maternal genes, that becomes unique. My cousins actually, they are dizygotic twins, okay, and one of them had identical twins. So talk about unique. So they're always asking questions. They're always wanting to do studies on him. Okay, so meiosis and mitosis, they have different purposes here. Mitosis is going to be replacing old cells or damaged cells, whereas meiosis is going to be giving us gametes. Gametes is the fancy word for sperm or egg. In mitosis, the result is going to be a cell that is identical to the parent cell, whereas the gametes have unique DNA. They have unique genetic combinations. They are different, yet they have similar processes. So we're still having interphase, we're still having prophase, metaphase, anaphase, telophase. Meiosis basically has 2 rounds of it. And then it also has what's called crossing over and independent assortment, which gives or contributes to genetic diversity. Did you read through that concept there? Now, errors can occur in meiosis if those chromosomes, those sister chromatids, if they fail to separate. So remember in anaphase 2, these should separate, okay? If this fails to separate, this whole pair goes that way and nothing goes this way. Or in metaphase 1, when the pairs line up, sometimes 2 can go over there and none will go over here. And so the failure of these chromosomes to separate, whether it occurs in meiosis 1 or meiosis 2 during anaphase, it's called non-disjunction. And what happens is you get an extra amount of chromosomes for that set. So you can see here in trisomy 21, tri means 3. Trisomy 21 is one of the non-disjunction events, or results I should say, that is compatible with life, meaning that they usually do survive, and they don't live a long life, but they do live into adulthood. And so you can see that right there. Down syndrome is specifically with the 21st chromosome. It's an extra copy in that. A lot of the other ones, there's ones called like Edwards syndrome, Patau syndrome. Some of them are compatible with life and some of them are not. Most of them are not. And so you can see right here in non-disjunction in anaphase 1, you can see that instead of this one going with that egg, both pairs went to one side. And so you can see you have no chromosomes on this. And so you can see right here, you have an extra set of chromosomes here with this sperm. Or when they fertilize, there's an extra set of chromosomes there. Here you can see on this one, there's only one set when there should have been 2 sets. This is showing after fertilization. When it occurs in anaphase 2, so non-disjunction can occur in anaphase 1 and anaphase 2. You can see that we have normal, you have 2 cells here, and then here we have this extra one, and here it's missing one after fertilization. So like we said, it can be lethal. Okay, so this one will be likely to survive. These ones are normal. This one is most likely unlikely to survive. Patau syndrome, this is chromosome number 13, which has a third chromosome there. Now, the sex cells are quite unique. So you can see right here when we get extra X's or extra Y's. You can see right here, these ones, Klinefelter and Turner syndrome, this one is, you can see the incidence of it. Klinefelter is a male, but it's got an extra X, so it's going to have more female-like characteristics, be infertile. Turner syndrome is a female. Jacob syndrome, they have an extra Y. Triplo X, they have an extra one of female right here. So this female right here, you do see Turner syndrome. They're considered a female, but they lack probably the ability of their ovaries to reproduce. And so I've just given you a couple examples here. So this is just showing Turner syndrome. This is a female that's missing an X, and you can see some of the issues that she may have. All right, and that is it for your Chapter 9 lecture.